and yam mosaic disease. What is the African Yam Project all about? African Yam Project was set up so that we can improve yam breeding techniques and develop yam breeding capacity among the national programs for accelerated development of high-yielding yam varieties with market-preferred traits, that is yam with very high quality. Breeding for resistance to mosaic and anthracnose disease is one of the objectives of the project. And the project covers 4 countries in West Africa: Ghana, Nigeria, Benin, and Cote d'Ivoire. The project is funded by Bill and Melinda Gates Foundation. What do we do in yam disease phenotyping in the Africa Yam Project? We are to develop and standardize high-throughput phenotyping project protocols for yam anthracnose disease and yam mosaic virus disease. We are to train researchers in partner countries, and then so that they can evaluate their land races and breeders' line for resistance to these 2 diseases. We need to first facilitate the development of marker-linked disease resistance using molecular tools, and to contribute to the breeding of elite disease-resistant varieties. So who are the members of the yam community of practice? It consists of all stakeholders in the Africa Yam Project and other yam initiatives. It is open to all categories of researchers working on yam, and is a forum for discussing and sharing protocols, standard operating procedures, general information. We share materials, and we train and carry out capacity development and other issues for the whole purpose of building a stronger yam breeding community. This webinar is therefore targeted at upgrading the skills in yam disease phenotyping. This leads me to the objectives of the webinar, which is to present the protocols that we are using for phenotyping for anthracnose and mosaic diseases, that is YAD and YMV. We are going to share recent updates on the disease phenotyping under the Africa Yam Project during the course of this webinar. We're going to present the protocols so that we can ensure that protocols are harmonized. We are going to discuss the way forward in this year and the next steps that they are to take. And our partners, what are we expecting from them in the area of planning, data collection, and analysis? And we are also going to identify areas that need clarification, because they are going to be also, they are also on the web now. They will phone in to ask their questions. We can phenotype in 2 main ways: by artificial inoculation or by natural field exposure. Both have their pros and cons. The advantages of phenotyping by artificial inoculation includes the fact that you can test under controlled conditions. You can control your conditions by yourself, and you can await a very reliable data, and you can assess your genotype reaction very well. However, the disadvantages include the fact that moderate resistant or recovery reactions are difficult to predict, particularly when dealing with fungal infections. And then you need to source for your inoculum, like the pure isolates of the pathogen that is causative for the disease. You need to prepare inoculum, and you risk wiping off all your germplasm, especially when you are doing whole plant assay. You always need a sick plot in the field so that you don't contaminate other field materials. And then it always requires field validation, so you don't rely only on the artificial inoculation. For the natural field conditions, the pros include that you need to prepare your inoculum, and then you can do it alongside other trials. While you are running other experiments, you can take data on diseases. However, the disadvantages are that the data are not always reliable due to heterogeneous infection rates within the field. And then there is variation depending on the reaction, depending on the location, the virulence of the prevalent strain, the time of infection, or climatic conditions. It requires retesting under artificial controlled conditions. So you can see that both of them go hand in hand. So the best phenotyping procedures combines both natural and artificial conditions. So we are going to talk more about the field phenotyping now. When planning for field evaluations, one of the things that you have to consider is your planting materials. What are you going to use? For yam, it can be mini sets. And when you are using mini sets, you must make sure that they are of equal sizes. And then they have to be pre-sprouted, and you transplant them later. And you can also make use of vine cuttings, which is the yam stem, and then, or botanical seeds, especially when you are working on mapping populations. For any proper good that you are using, it is important that you plant on the same day so that you can have uniformity. As for trial design, you can use any design, but the right spacing is essential so that you avoid contamination on field. And then at least 3 replications should be used so that you can have good data that will be analyzable. Then you need spreader rows, that is susceptible checks that will be on the field that will be able to introduce the inoculum and also help to assess the level of the natural infestation, because as is the level of the infestation you see on the susceptible checks that will be able to tell you whether there is a right inoculum on the field. Another aspect of the planning is timing of data collection. It is very important. Depending on the start of the rains or the availability of irrigation facilities, it can either be in April or May, based on yam physiological requirements. And then you score at 3 months after planting, 6 months after planting, or at 9 months after planting, or at harvest. You can follow the same schedule for both anthracnose and mosaic virus. And it's also essential that you observe at 1 month after planting, especially for the virus disease, so that you can recognize tuber-borne disease or seed-borne diseases if you are using botanical seeds. For the data collection, you collect data on disease severity and on incidence on the specified dates. And like I said earlier, we need laboratory validation to confirm virus and the fungi, making use of plate assays, ELISA, that is enzyme-linked immunosorbent assay, and PCR, polymerase chain reaction. Very, very important, you should be able to take photographic records, because you can always go back to this, especially for the digital imaging that we are encouraging now. You'll be able to score your severity based on the photographic records that you have taken. This is a format that we are suggesting for data collection. It may vary depending on what you really want to achieve, but you must have the essential details in it. You have the date of planting, your date of scoring, and other relevant data like location, the GPS coordinates, or even the name of the person that is scoring. I just want to chip in now that it's good to have the same set of people scoring the same fields to avoid bias in our records. Then we need to have the name of the accession, of course, the disease that we are scoring, and the plant number so that we can assess the total number of plants and the number of infected plants later, and we'll be able to now calculate the mean severity and the incidence later. It is important to take the data on the specified dates, because we are working on putting all our data in a repository in the YAM base, and we expect that everybody will take their data at the same time so that we can be able to have uniformity in the YAM base, and we'll be able to analyze our results together. And we are also encouraged to use the Fieldbook app, which is an electronic device for data capture. It avoids the drudgery of having to enter your data yourself after coming back from the field, and therefore minimizes error. There are scales that we use for scoring on the field. For yam anthracnose disease, we have a scale of scoring of 1 to 5. The plants are assessed based on the symptom severity. The score 1 has no symptoms, while score 5 is almost dying. The score 2, 3, and 4 are somewhere in between. I'm still going to explain the percentages in another slide. This also goes for the yam mosaic virus, with the scale of scoring of 1 to 5 also, 1 being the one with no symptoms, and 5 being the most severe. Now we have an harmonized scoring scale for both diseases, whereby you can give a kind of rating or classification based on the symptoms that you are getting. For highly resistant materials, they are the ones that we give score 1, and those are the ones that are showing no symptoms. The resistant ones have percentages between 1 and 25% of the total plant area infected. Moderately resistant plants, which we give score 3, have between 25 and 50% infected area. The susceptible ones, 50 to 75, and highly susceptible materials between 75 and 100%. Those are the plants that are almost dying. For our data analysis, we calculate the incidence as the percentage of the number of infected plants over the total number of plants. And for the severity, is the percentage of the damaged leaf or plant area over the total plant or leaf area. We can also calculate the area under disease progression curve, making use of the data on severity over a period of time. This slide is showing us the formula used to calculate the AUDPC, and this can be adapted in Excel. And we are also working on how to get it into our YAM base so that it can be calculated directly from the YAM base after entering the data. So talking about the protocols that we use now. For YMV screening, we have been using mechanical inoculation, and we found out that it is aiding us in having 40 to 90% success. And then for yam anthracnose disease screening, we are making use of detached leaf assay. But the additional protocol we are using is scoring with digital images, making use of an app called Leaf Doctor. This app was developed by Petribridge et al. In 2015, and this can help to score accurately the leaf, percentage leaf area damage of our leaf samples. So I'm going to talk about the lab or the screenhouse-based evaluations now. When you are planning for your lab-based evaluations, you have to consider the source of your inoculum. You need to have pure isolates derived from YMV-infected accessions from each country. Why we are doing this is that we don't want to exchange diseases between the countries, so each country must isolate their own pathogen. And then we need pure isolates for the yam anthracnose causative organism, which is Colectotrichum gliosporoides, from both materials having the spot and the blight symptoms from each country. Then we need to culture our pathogen. We need to prepare the media and get other lab equipment ready and lab wares ready. And for the YMV, where we need to culture on the host plants, we need to plan the planting of our host plants to coincide when they will be ready for to coincide with the time we want to do inoculation. And we require expertise of good personnel that are trained, that can oversee the experiments. So basically, we need some, there are some requirements for the screening of yam anthracnose disease and yam mosaic virus. We need the virulent isolate for whatever we are screening. We need facilities for inoculum preparation. We need to have susceptible accessions to use as controls, because that will enable us to be able to compare with whatever we are, our test samples, to know whether they are actually resistant or susceptible. We need good screen houses so that insects will not enter to act as vectors for transmitting diseases. And then we need the right environment, that is the right temperature and humidity for culture growth and disease development. There are specific needs for yam mosaic virus screening. You need healthy plant materials as starting material. And then the hemp bentamiana is required as host for the mechanical rub inoculation. And you also need the facilities for the enzyme-linked immunosorbent acid ELISA that I mentioned earlier, and polymerase-based detection, polymerase chain reaction-based detection. So now I'm going to run through the protocol we are using for yam mosaic virus screening. That is the mechanical inoculation method. We culture our YMV first on Nicotiana bentamiana, and then we prepare our inoculum now by grinding in inoculation buffer. The inoculation buffer is, it consists of phosphate buffer with an additive known as diaca and abrasive that can cause damage, help to cause the damage on the leaf surface to enable the virus to penetrate. This time we are using cellite. You can also use carborundum. We apply the inoculum on the yam leaves by rubbing it gently on the leaf surface with our hands, wearing gloves. This protocol had been given to all our partners, so they have hard copies of the protocol and the soft copies also. So we do this inoculation at the 3-leaf stage or 3 weeks after planting, when the plants are ready to be inoculated. And then we rinse off after the mechanical application to rinse off the excess inoculum. Then we leave to grow and watch out for symptoms. This we score on weekly basis. And it's also very good to always have on inoculated controls, both for your test samples and the host plants, the Nicotiana benthamiana, so that you can be able to make good inferences later. For the yam anthracnose disease, the protocol that we are using for the detached leaf assay or the whole plant assay involves us having the virulent isolate of the causative organism, Colectotrichum gliosporidis. We need all our test samples, and we also need to culture the pathogen. So we should culture this 7 to 8 days, after which the inoculum will be prepared, and then we adjust it to 1 million spores per mil, which is the recommended concentration for inoculation. From these pictures, you can see that Colectotrichum gliosporidis is a very diverse organism. Then we need to harvest 3 leaves per plant from the top, the middle, and the bottom, and then 2 plants per session. We surface sterilize this and then place them face down on the filter paper moistened with sterile disease water. We inoculate on this Babesia surface because we have more stomata on that side of the leaf. And we apply 4 drops of 10 microliter each to this surface of the leaf, and then we incubate at 28 degrees C and score at 7 days after inoculation and 15 days after inoculation. We need to take digital images of these leaf samples at each time of scoring, because our leaf doctor will work on those images. This is showing us the workflow of the detached leaf assay. You can see the surface sterilization being done, and then the plating of the inoculation, plating of the leaves and the inoculation, after which we seal the plates and incubate. And then after the incubation, we do post-incubation assessment, which involves the taking of the pictures from which we'll take the data. We must make sure we are using very good cameras that has red and green and blue spectrum, and we must have uniform images that as regards to exposure to light, distance from focus, and pixels, so that we can be able to compare whatever we are scoring. We need a background for the analysis when we are using this app, and therefore we can adjust the background of pictures using photo editors like Magix and Adobe. Then we need color charts and rulers for standardization and comparison. For the whole plant assay, we can do it simultaneously with the detached leaf assay, because we can the leaves could have been detached from those same plants that we're going to use for whole plant assay. These plants are sprayed with inoculum, making use of a sprayer, and then we cover the plants with nylon bags to maintain high humidity. And it's also essential to make sure that the plants are wet before the inoculation, so as to also increase the humidity, because the spores grow best in humid conditions. And then we need to observe symptoms as from 1 week after inoculation, and our data collection will be based on this. So what are our next steps for 2017? Our partners are going to follow this protocol for screening yam mosaic virus disease. They need to identify the yam mosaic virus resistant materials that were identified from last season or the previous season. If they are going to make use of vine cuttings, you should make their vine cuttings very early in the season before the plants start to flower, because at that time, you are sure, at this time, you are sure that they are going to grow well, depending on the genotypes. Then we plant the proper goods in the screenhouse at about 10 or 20 cuttings per session, and then we make use of optimized protocol as suggested by Otu et al., in which you can use 3 parts of soil with 1 part of carbonized rice husk. He has tried it and says that it's working. And for us too, we are making use of 50% soil and 50% substrate, and that is also working for us. And you index whatever you are going to you want to test to confirm that they are virus-free. Because whatever you want to be sure that whatever you are seeing as symptoms later is as a result of your inoculation. And then the next step is to validate the ones that are that are virus-free by doing mechanical inoculation, and then we will index later to find out which ones are susceptible and which ones are resistant. Then, after we have been able to do this, we can nominate accessions to breeders so that they can revalidate in multi-site testing. While doing this also, we still continue with our field screening of our germplasm. So what do we expect from our partners in the collaborating countries for VMB resistance screening? We need to make a screening plan based on recommended protocols. And we need to carry out our detailed screening of germplasm under natural field conditions. And we need to extract nucleic acid for virus testing so that we can do qualitative and quantitative assays. For the yam anthracnose disease resistance screening, both for the lab and the screenhouse, they also need to select resistant accessions from the previous season and make cuttings very early in the planting season if they are making use of vine cuttings, and then plant the proper goods early enough and allow them to grow. Then we carry out the detached leaf assay with the aid of the digital imaging leaf doctor app that I told you about. And then we validate by whole plant assay in the screenhouse. From here also, we can nominate accessions for revalidation and multi-site testing. We also continue with natural field infestation for all the germplasm. Then what do we expect from partners in this case also? They're supposed to also make a plan based on our recommended protocol. They are supposed to perform detailed screening of the germplasm and also follow the pipeline that is recommended. In summary, I would like to say that proper planning is needed for both field-based and lab-based experiment, that is artificial inoculation and the field phenotyping. And we should always realize that we have a very short time frame to take our data, so we must plan our work very well to coincide with the yam planting season. And then we have the subjective data that can be optimized with the use of digital phenotyping methods. Because when individuals take data just by visual observation, it's error prone. But if we can be able to use digital imaging, we get better and accurate results. And the use of electronic data capture devices are encouraged. All the partners have tablets that they use in collecting their data in the different countries. And then we should have it at the back of our minds that a good phenotyping platform is the backbone of any genotypic research. With this, I would like to say thank you very much.
You said that the mini sets need to be pre-sprouted. Is it compulsory, or we can do it directly as mini sets?
If you're going to do it in the screenhouse, if you it's good to transplant it because we do it in a big bowl to have maximum number that you are pre-sprouting, and they grow at different times. So it's good to pre-sprout so that you can have material with at the similar stage that you are going to inoculate.
Thank you. long has the YAM community of practice existed? That's the first question. And then also to find out, you know, you had your scoring methods and you want to ensure, you know, standardization. Have you shared those scoring method with them, said that it's like a card they can take to the field and use? And have you also considered hotspots, you know, to ensure spread of these diseases? Thank you. Thank you, sir.
Thank you very much for your question. For the YAM community of practice, I think it has always been there, but not as a coordinated community. But with this project, the inauguration took place in Tanzania when we were having the IICRC, just before the IICRC AB meeting, where most of the people involved with YAM research were around, and we had the inauguration meeting. And now we have our website at the Africa YAM website, so that we can communicate and exchange ideas within ourselves, among ourselves. Pertaining to the protocols, we organized training here sometimes last year, I think around this time last year, before they started the phenotyping in their various countries, where we shared these protocols with them. And when they were going, they went with both the soft copy and the hard copy of the protocol, and so they can follow and do all this on their own. And as for the hotspots, one of the things that we did at the beginning of the project was to send like a questionnaire to all the partner countries, and they helped us to identify hotspots for all these diseases in each of the countries. And so we know the hotspots in all the partner countries where we can do the validation.
Yeah, thank you for the presentation, clear presentation. My question is, you said you have your partners in different countries use electronic data capturing devices. How do you run it decentralized, or you have a central repository where all the countries will send their data and you put it in a central place? That is one. And then secondly, I'm sure you must have heard about open access.
Yes.
Is those your data being organized and in a open access format and compliant?
Yes.
Including the ontologies?
Yes. We have a YAM-based, how will I say now? A YAM-based site, or people that are working on YAM-based. YAM-based is like a repository where everybody keeps the data from the different, it's not only for disease phenotyping. Everything you want to know about YAM should be there. And we are trying to make it such that it can do even data analysis, and everybody that wants to know something about a particular YAM variety can always go there and maybe put the name of the variety, and you get all the information that you need to know. And you can even plan crosses and do different things about breeding in making use of that YAM base. We have somebody that is in charge, Dr. Agre and Afolabi Agbono, both of them. And then we have some partners also who are trying to make sure that this YAM base is working well and it's compliant with the open access technique.
Yeah, thank you for your presentation.
Yes,
you said when you use mini sets, you have to score 1 month after sprouting to know if there is some viruses, or imagine now the virus you want to spread is already in the mini set. How you evaluate what is there before and what you are bringing?
Usually for our screening work, what we do is, when it is in the screen house, we grow it out and see how the symptoms are coming. So we are not going to treat the ones, we are not screening the ones that are already showing the symptoms again. We know that they have the virus already. So the ones we want to screen are the ones that seem symptomless, and yet we want to know if they are actually resistant or not.
So thank you for a nice presentation. I have 3 questions. One, do you have any recommendation recommendation, particularly for virus race that is really highly virulent out of many races, because so that people can go only for that virulent race for screening in the natural condition or in the lab condition. For both anthracnose or maybe the mojivirus, particular race. Okay, this is the race really very virulent, because in the pathogen there are many races, some are less virulent, some are highly virulent. And question number 2, as you said, we can screen after 3 months, 4 months. I agree. Do you have any recommendation? Because in the yam, of course, there will be some early maturity varieties, some will be very late maturity variety. If you are planting both together and you are going to screen them in after 3 months, then means little bit bias here, because maturity period or maybe the growth rate is not same. Because late period will go late, so I mean we are not giving the same time. Yeah, so maybe we have to look for the maturity period, and then we have to plan, okay, this is the maturity period, so in one group, and early maturity period in one group, and then we are giving our infestation. And third is just for my lab screening, you are saying you are taking from the 3 leaf, up, middle and bottom. In the bottom, in the 3 months, of course, in the yam, it is the creeping crop. Maybe the bottom is not getting the, it is inside when we do, or maybe because of the senescence or something. So do you find any effect whenever you just look only the bottom one, it is highly susceptible, and if you go up to the reading is little, the scoring is coming little less something. So it make any biasness in the reading or not?
Okay, maybe I should start from that. Well, we are taking from the 3 different parts so as to avoid this bias, because we know that the bottom one is even closer to the soil, and then with the splattering of the rain, they are likely to get more infected or more moisture than the ones up. And then maybe because of the size, especially when you are doing old plant assay, it may be that the smaller ones are more susceptible because they are smaller than the bigger ones. That's why we are taking at the different points so that we can avoid the bias. For the maturity period, you have a good point there. I think it should be part of the planning so that we can have the early maturing together and the late maturing together. But what is important, we must take it at the specified period. That means our 3 months after planting must be 3 months after planting so that we can compare our results. And then for the particular race, we have the viruses for us in air that we are already keeping, and we know that they are virulent, and we already have the sequences so that we can always check if it's the same one that we are using that is manifesting or not. And then we have told all our partner countries also to isolate the most virulent materials where they are. Those are the materials, the pathogens they will be using for their materials. And here too we have already backup with the pathology at the germplasm health unit where we do the yam anthracnose disease screening.
Let me add to the issue of scoring on top, middle and bottom. Yam is a crop which keeps producing new vines. So when you score later at the bottom, it is not necessarily old leaf. You're actually looking for the new vines that comes in. And quite often for viruses, at the early stage sometimes they don't show any symptoms. But often it is the new vines that come later on, they show symptoms. And this happens either because of infection later stage of the growth, and sometimes in some genotypes they have some kind of recovery, they don't show symptoms at the beginning, but the minor new vines that comes out later, they show. So this is a deliberate strategy, particularly for viruses. For anthracnose, this is mainly done when we do the detached leaf. As we take 3 positions to make sure that the position of the leaf is not affecting the spread of the rate of spread of the pathogen on the leaf. Okay, in the field conditions, usually anthracnose, if it is a spot type, you pretty much see at the lower leaf, you don't see much on the top. And if it is blight, you pretty much get a systemic one day back. So it doesn't make much sense to see the whole top, middle and bottom in such situations.
No, actually why we use this, okay, suppose in the bottom, yeah, because of the soil, maybe our leaf has the most moisture is there. So maybe the leaf on the top is coming in the category of 2. But in middle it is coming 3, and we are coming in the bottom, it is coming in the 5, and in the last, if you will look only for the bottom one, yeah, on the rest one it is, oh, this is going as susceptible, and it's Because in the last you are going to take the mean bottom and this in the mean scoring in the AUD basis. So maybe it has little biasness, something. I think that's right.
Actually, I want to have a follow-up on the same thing, on the scoring itself, the scale of 1 to 5. You know, today what you are scoring as 1 may become 3, and I know that it is under the AUDPC that you cover everything, but do you actually use a couple of people, you know, because what you will score as 2, I may score it as 1. Now there is a human bias, how we look at it. That's 1. Yeah? And 1 to 5 scale is always a kind of a little biased in that situation. That's my first question. The second question, maybe Laura can respond. If I have to do, if you inoculate today, how soon do you see the reaction? If I have to do some RNA sequencing, and I want to understand the host-pathogen relationship. Thank you.
Okay, thank you very much for your question. For the rater's bias, what we try to do is to, usually we score together at the beginning, if I have people that will have to score with me. So I know the way they score. As in, we score together, we all decide on, okay, this is 1 or this is 2 or this is 3. And by the time we have been doing that over and over again, I know that's why it's digital imaging that is actually the best. But for that, we can still be able to say we are harmonizing our scoring because we already know what we want to assess. We do it based on the severity and not just what you just want to pick. Most of the time, we have found out that we have similar ways of scoring. That's why you need experienced raters to do the rating for you.
Ideally, you should use only one rater for the whole, so the bias is normalized.
Yeah.
If you use different people, then it becomes more messy.
Yeah.
And then the RNA-seq, there isn't any hard and fast rule. It all depends on how much money you have because there is no.
No, but what stage? When if you inoculate it today, so should I take the sample after 24 hours, or I should do it? I know it should be a periodical, 24 hours, 72 hours, like that.
If you are looking for hypersensitive response genes, which we should be taking 24 hours, or even followers 24 hours, because these genes are the ones which trigger cell death. But if you are looking at much more systemic response, then you need much more.
So what is the much more? It's 1 week, 2 weeks?
Depends.
For virus, for example, it's different because it has a slow reaction. Whereas fungi, it depends on the kind of host pathogen response, and accordingly you need to have. Anthracnosis is not a very readily systemic pathogen. So when you, this is something that we have to do a separate experiment, wherein if you inoculate on one leaf, would it trigger resistance response in the upper leaves, whether the susceptibility reduces. Okay, these kind of experiments need to be done. But even to do that, we need to identify genotypes to demonstrate that kind of response exists. So right now we don't have that knowledge. All we are doing is, that's why last year we have taken top, middle and bottom leaves to see whether we get any differential response, but unfortunately the data was not clear enough to make any conclusion. So we still need to have a fundamental understanding of the host response to some of the pathogens to design a proper RNA-seq experiment. But otherwise, to start, anything is good.
Yeah, but we have now some plants which are growing. Ibukun, you may explain more. We have some of the parents that we call as resistant. We don't know whether they are really resistant, but we think that they are resistant based on our scoring and whatever. And then we have about 7 parents, of which we have some resistant as well as some susceptible. And I think we have the funds to start
I think
something
the plant
like
numbers
that.
are there are not too many plants. We need more plants. Then we can design different experiments just to understand, okay, by inoculating only lower leaves and then see the spread of the pathogen, and then subsequently we can check, inoculate on the further leaves to see whether the response of the host is different compared to the lower leaf. So we need more plants. Right now we have 10 plants, which is too small.
So we can do fine cutting. Okay, let's discuss this further.
Okay. I want to find out for the digital imaging. It's the means that we can eliminate bias, reduce bias. But do you also do that for field phenotyping? Now that you have to detach your leaves, so how do you do that when you want to do field phenotyping? Can the digital imaging also be used, or it's only when you have a spin-off?
It can be used, but we don't have the facilities yet.
Yes,
because like we can use spectral imaging for field-based assessment. In that regard.
Thank you for your presentation. My question is on the detached leaf assay. I don't know if one wants to carry out this sort of experiment, maybe one of your partners. I don't know how many leaves one is expected to get from a particular plant, for example, to assess in the lab. And if there are multiple leaves from a particular plant, how much variation should I expect? If I inoculate leaf A and I inoculate leaf B and I want to compare, how much variation would I get in a particular plant and in different plants from the same accession?
Yeah, just like Dr. Lawa was saying just now, we tried to do that last year, but we were not able to get concrete evidence to say, okay, this is the best way to do it or not. But from what I can see from the data leaf assay that we did, for some materials that are the same genotype, all the 3 leaves behave the same way, and for some, you see them reacting differently. So it's something we still need to work on to be sure of what is happening. And we always take 3 leaves per plant and 2 plants per accession, depending on the number of material that we have.
Yeah, just I have a quick one here. Maybe I'll be asking one for myself or the GHU. For the YAD and YMV scoring, we have 3, 6, and 9. I'm a little bit confused with 9 because 9 is most of the plants.
Yeah.
And when he is going to come to my field, for example, and it's likely to come once for inspection, at what age of the plant can we say it's most suitable for YAD and for YMV assessment?
That's fine.
Okay.
Why we do at 9 months? This 9 months is particularly for virus. When the plant show recovery, so you would see that is the most latest you can see. Yes, it is close to senescence, but that's the whole point. For anthracnose, it's also good to do at 9 months. If it has a spot reaction, if it is not expanding, so yam starts like it goes through wet period and then 9 months is a dry season. Okay, if it is not expanding, the spots remain as it is, and some leaves may drop off, but the plants are wet. Whereas if it's a blight, the whole plant will die within 3 months when it gets infected. So you don't have to go up to 9 months. So depending on the crop, you need to go up to 9 months. But for the GUT inspection is different from the phenotype. GUT inspection is mainly to do with whether the field has any quarantine pathogen. And ideally they do between 3 to 6 months before senescence. Okay. Between 3 to 6. There has to be good enough foliage and there has to be enough exposure for the plants in the field.
Sorry, and lastly, if for the detached leaf assay, it suggests to me that the leaf in the petri as shown, for example, when inoculated with CG is likely to be more affected than while it's on the field because the leaf is already detached from the soil and it's expected to move. So the data I have from that, how do I balance it?
There has been some work on that before, and the person that did the work reported that there was correlation between what happened for detached leaf assay and what happened at the screenhouse field level for yam. I know there's always arguments on different crops for that, but we have data to back ours up.
Okay, see, the leaf assay is a kind of rapid phenotype. and get a quick feel of how is the plant behaving. And whatever you observe in the detached leaf assay, you need to again reconfirm by going for whole plant inoculation. And this has to be done under greenhouse conditions, not in the field. So the previous study has shown a good correlation between the observations made in the detached leaf versus whole plant assay. So that way, the real method is kind of a quick screen. If you have hundreds and thousands of samples, you quickly can narrow down to small set, and then you go for intensive phenotyping on those small sets. Yeah. You can't conclude, like that is, again, once you do the detached leaf and again the greenhouse, that is not still the end. You still have to go for multi-site evaluation, particularly for anthraquinones, because the pathogen is highly variable. So we need to take it to different locations to validate, and then only we can conclude whether it has broad spectrum resistance or specific resistance to particular pathogens. Good. If there are no further questions, thank you very much for coming. This is a kind of experiment. It's an experiment. It's a webinar. Supposed to be just, the speaker is supposed to just speak to the camera and then later on telecast this one. But somehow the announcement came as a seminar. But I really appreciate everybody coming here and the nice questions. Yeah. Thank you.